Hey everybody, we're back for lecture number 2 on the brain. We are still in the frontal lobe. You can see this big, beautiful brain right here. We're still in the frontal lobe. And let's draw on our frontal lobe just for a second here. Still in the frontal lobe. Boom, boom, boom. There it is. So we've done the primary motor cortex, the precentral gyrus, in other words. We did the premotor area as well. And now we're going to do the motor speech area. The motor speech area, also known as Broca's area. It's called Broca's area because of this handsome guy right here, Mr. Broca. I don't know his first name. Probably Johnny. That's my guess, Johnny Broca. I don't know, but I do know that his area is right here, this dotted line area here. That little area right there, that is the Broca's area or the motor speech area. If we look at that word, okay, motor speech, movement for speech. So this area is going to control muscles. There's the motor part. Muscles for your tongue, lips, and your throat, your pharynx, in other words, that you are using to talk. I am using my motor speech area right now because I am talking to you. It is involved in making the movements of your lips, of your tongue, of your palate, of your pharynx. There's really a lot to speaking when you think about it. And this area is actually super interesting because it is one of these things where we find it on the left side of the brain. So it's in the left hemisphere. So that's normally for most people. For most people, the dominant hemisphere is the left hemisphere. Whether you're right-handed or left-handed, for most people, the dominant hemisphere is the left hemisphere. And it's on the left. It's not on the right, which is really super interesting. Okay. That's Broca's area. Don't want to damage your Broca's area. Definitely not. All right, let's keep on going. Let's talk about the prefrontal cortex. The prefrontal cortex is the anterior region, the frontal lobe, but in the front. I think of the prefrontal cortex as the frontest part of the frontal lobe. Frontest frontal lobe. This is where we do a lot of the things we really think about as brain stuff. This is where we're analyzing things. We're doing cognition, thinking. This is where we have our personality, where we have our conscience, where we control our impulses and our behavior. This is the part of the brain that really doesn't fully mature until you are in your 20s. My dad used to say to me, "What were you thinking?" when I did something dumb. I should have told him that my prefrontal cortex wasn't fully developed yet. And he shouldn't get mad at me. I don't know if he would have bought that. Just like you're not going to tell me that you did poorly on an exam because your prefrontal cortex still needs some developmental time. All right. Yeah, different animal species, by the way, have different sizes to their prefrontal cortex. So you're going to see that if you look at some, dumb animals will tend to have a smaller prefrontal cortex. I don't know what's bigger, a dog or a cat. I wonder. I'm going to look at a picture of it. You know what? It looks like the dog has a bigger prefrontal cortex than the cat. So, that's good. I like dogs. Don't like cats. Don't know if any of you are cat people out there. Cats are pretty useless in my point of view. Anyhow, let's keep going. Oh, yeah. Have you ever heard the story? This is a great story. This is the guy named Phineas Gage. Phineas Gage. So the year was 1848. I might be off by a year or two, but I think it was 1848. The year was 1848. He was a railroad foreman. He was a hard-working guy, smart guy, diligent, courteous, respectable, nothing but good adjectives in front of this guy. Okay? And what he was doing one day is they were building a railroad, and he was, I believe he was tamping down some gunpowder. So he was using an iron bar to pack gunpowder into area of the rock where they were about to try and get it to explode and to get some rock out of the way to build that track. And he was using a big iron rod, and somehow iron rod hits rock, spark happens, and gunpowder explodes. The iron rod shoots up through his face, through his eyeball, comes out of the top of his skull up here. Crazy, right? He didn't even lose consciousness. They put him in a wheelbarrow, wheeled him to town, brought him to the doctor. Somehow the doctor took out the iron bar, kind of like tried to put the bony pieces back together, like put the jigsaw puzzle back together, bandaged him up. And amazingly, he survived. He didn't get an infection, he didn't die. But as the story goes, he went from being this incredibly nice, respectable, conscientious, diligent guy to almost the complete opposite. Someone who was whose personality was, would be like a total jerk. And so that's the way the story goes. I don't know if it's totally accurate, but it's a good story. And you can see what's going on here is the iron bar went up through what lobe of his brain? If you're shouting it out right now, then that is good. The frontal lobe. So, pretty interesting. Back in the day, people used to get what was called a frontal lobotomy, where the frontal lobe would basically be cut up with an ice pick in an effort to cure, and I'm putting air quotes in the word cure there, to cure things like schizophrenia and other mental disorders. All right, on that note, guys, we are done with the frontal lobe. Hooray. We did the primary motor cortex, the premotor area, the motor speech area, and the prefrontal cortex. There's more parts to it. There's a lot more. We are just doing on like a greatest hits of the brain here, a highlights tour of the brain. And we're now moving to the parietal lobe. We are in the postcentral gyrus. Right here is my central sulcus. So this blue guy here is my postcentral gyrus. It is the primary, so first, most important, somatosensory cortex. Somato, body. So we're sensing the body here. We're going to be aware of things like touch and proprioception. And this is also a contralateral situation where the left side of the body, if I smack my left hand here, I feel that, quote unquote, in the right side of my brain. So there is contralateral input for this. So this is our primary somatosensory cortex right here in the postcentral gyrus in the front part of the parietal lobe. If we look, we can see the same arrangement, I shouldn't say the same, a similar arrangement to what we had within the precentral gyrus. Again, we have body parts that get brain space devoted to them. This time, it's not based on how much movement they're capable of, but on how sensitive they are. How sensitive they are to things like touch and proprioception. Notice that if we look, some areas like the hand and the fingers are still big. Tongue is still big. Lips still big. The eyes got smaller. That's a big difference, isn't it? There's gums. You didn't really have a motor area for your gums before. Genitals. You're not going to have the same degree of movement at the genitals as you would in terms of their sensitivity to touch. Plus, there's an intra-abdominal region as well. If we look at this guy, he's definitely similar looking. He's the sensory homunculus. We had the motor homunculus earlier. He's definitely similar looking, but there are some differences. All right. Just posterior to that postcentral gyrus, we have this region called the somatosensory association cortex. This is where we try and figure out what's actually touching our fingers, for example. Let's say you reached into your pocket and you were like, whoa, it's full of money. And you had all this money in your pocket right down here. Could you tell how much it was without taking out and looking at it? Could you count it and be like, all right, I got 2 quarters, I got a dime, I got a penny, I got a nickel. Like, could you feel Thomas Jefferson's profile and know it was a nickel? Feel FDR's profile on the dime, Washington on the quarter, Lincoln on the penny. Could you feel those and figure it out? Maybe by size, that might be, and the ridges and things like that. But you could figure out what you're touching, what's touching you, and you could know what the object is, or have a better idea of what the object is. So you're getting a better idea of what the object that's touching you actually is, what it means, what its importance is in this somatosensory association cortex. All right. Done with the parietal lobe. Zipping our way into the temporal lobe. In the temporal lobe, right up here, near that lateral sulcus, so the superior margin of the temporal lobe, we're going to have our primary auditory cortex. So sound waves are shaking your ossicles in your ear. You shake the eardrum, you shake the ossicles. They shake something called the cochlea, which then creates a nervous signal, which travels and gets to this auditory cortex. This is where you first start to make sense of the sounds that you're hearing in this primary auditory cortex, which is located in this portion of your temporal lobe. And again, guys, remember we're doing just the greatest hits of the brain here. So we're not going to do a ton of temporal lobe. There's another part that's in the temporal lobe, most of it at least. This is called Wernicke's area. This is an area that is involved in understanding written and spoken language. So it's one thing to hear me. It's another thing to understand the words I'm saying and put them together and comprehend them. And that is going to depend crucially on Wernicke's area. And we see him. He's got a nice beard down there. All right. Now, if we kind of peel the lateral sulcus open, just on the inside of that temporal lobe right there, the medial temporal lobe right there, that is our olfactory cortex. Now, you know you have olfactory nerves in your nose that are going to smell smells. Signals go up through that curviform plate of your ethmoid bone and onward to the olfactory cortex in the temporal lobe. Now, what's super interesting about the olfactory cortex is that it also plays a role in memory and in emotions as well. I always thought that was pretty neat. That smells can arouse a certain memory. Okay. Let's keep going, guys. In the occipital lobe, in the backmost part, so like the very back tip right here, is the primary visual cortex. So obviously, right now, light is bouncing off things wherever you happen to be. Light bounces off things. Light enters your eye, hits your rod cells and your cone cells and your retina. Signals eventually make their way all the way to the other side of the brain, to the primary visual cortex. So this is the part of the brain where we first sense light. It is the biggest of our sensory cortices. So you got bigger cortex for vision than you do for smell or hearing or touch. Because we are really reliant upon vision. We really are. Now, there's also, here we see the pathway, which is pretty neat. And I almost forgot to tell you this, so I'm so glad I had this picture here. In this pathway, there is a contralateral situation going on, but it's weird. Earlier we said touch was contralateral, right? I touch my left arm. It's my right parietal lobe that's getting that info. Vision is a little bit different. It is not left eye, right brain. Right eye, left brain. That would be too easy, okay? What it is field of view, okay? So like, if you're looking straight ahead right now, you have a huge field of view, okay? Huge field of view ahead of you. And imagine drawing a line straight out from your nose, like straight out forward, okay? And everything to the right of that line, that's your right field of view. Everything to the left of that line is your left field of view. And so what's in your right field of view goes to your left occipital lobe. What's in your left field of view goes to your right occipital lobe. All right? So each field of view will involve both eyes. But that's the contralateral way vision works. And you can see it. Like, look at how when the axons come out of the back of the eye here, the retina, only some of them cross over, okay? Only some of them cross over. All right? So the consequence of this is that our visual fields are contralateral. All right. There's also a visual association area, which kind of like surrounds the primary visual cortex. This is where we figure out what we're actually looking at. Think about the primary visual cortex is just saying light. There's light hitting my eyes. There's light hitting my eyes. The visual association area is where we figure out what exactly are we looking at. What is this combination of light and shadow that we see? Is it a person? Is it a person's face? Is it a person's face? Did I know that person? Etc., etc., etc. All right. Excuse me. Let's keep going. We're doing great here today, aren't we? Let's talk about that insula for a second. That's the hidden lobe, the island lobe. Insula means island. You know, like peninsula is almost an island, right? So insula is an island. It's tucked back in there. The insula does a lot of things. One of the major senses is gustation. It's got your gustatory cortex. So that is your sense of taste. It's also involved in other things. It's involved in interoception. Let me write that word down. I haven't done much writing on this particular video. Interoception. This is your sensation of things like stomach fullness, bladder fullness, things like that. That's your insula. Your insula is also involved in some emotions. It's very much involved in the idea of disgust in both senses of the word, like something tasting disgusting and also something being like morally disgusting. Your insula plays a role in both of them. Okay. So with that, we're done the insula, and we're done with our walk through the lobes. What we now got to do is look at some of the nuclei. So remember, earlier we had said the outside of the cerebrum was gray matter, which is the cortex. Then we had white matter, which is myelinated axons. But then there are other regions of gray matter that are deep to the cortex. And these other regions of gray matter that are deep to the cortex, they are known as basal nuclei. Basal because they're deep. Some of them are involved in planning movements, making sure movements are smooth. like deciding when to start and stop movements. The ones that are involved in that are the guys that are kind of more superior and more medial in our picture here. These three right here. There's one called the caudate nucleus, one called the putamen, and one called the globus pallidus. And these two guys make up something called the lentiform nucleus, and all three of them make up something called the corpus striatum. Another one is called the amygdala. This little guy down here is the amygdala. Amygdala, I think, means almond. Like, it looks like an almond. It is very much involved in your emotions and in your moods. What if you didn't have a functioning amygdala? That would be crazy, isn't it? Your amygdala can get damaged. Basically, your sense of fear would be greatly reduced if you did not have an amygdala. I guess it's kind of cool. Kind of bad though. I mean, fear can be a very informative thing. Okay. Anyhow, these are our basal nuclei that we have to be aware of. And now we're done with all the gray matter in the brain. Not in the brain, sorry, in the cerebrum. And we got to introduce ourselves to the three varieties of white matter in the cerebrum. All right? Three types. There are fibers called commissural fibers. There are fibers called commissural fibers. There's that word right there, commissural fibers. Commissural fibers will connect the left and right cerebral hemispheres to one another. So right here, this is in green here, this is the corpus callosum, which is a great example of commissural fibers. So commissural fibers connect the two hemispheres to one another. Then we have association fibers. Association fibers are going to connect one part of a hemisphere to another part of the same hemisphere. Obviously, these different cortices have to talk to one another. I mean, the auditory cortex and the visual cortex have to talk to each other. So when you see someone and you hear their voice, you are recognizing and remembering who they are, right? Cortices have to talk together to do this. And the association fibers let you do this. Lastly, we got projection fibers, the purple ones in here. We already looked at an example of projection fibers, the pyramidal neurons that come out of the prefrontal gyrus and go down towards the spinal cord for movement. We talked about how they do that decussation. We already talked about that. So projection fibers are going to go from our cerebrum down towards lower structures like the spinal cord. So three basic types: commissural, association, and projection. Okay. Good. Let's erase all that. Let's look at another brain picture here. If we look at this actual brain, we can see projection fibers right here. See projection fibers right here. We can see commissural fibers right here. And then these guys over here would be association fibers. Cool. All right. Here we see left hemisphere and right hemisphere. I see all of these bundles of axons uniting the two hemispheres. So obviously, what type of fibers are these? Are they commissural? Are they association? Or are they projection? Well, if they are uniting, connecting the two hemispheres, then by golly, they are commissural fibers. All right. Good stuff, guys. Hey, hooray. Hip, hip, hooray. We are done the cerebrum. Which means all we got to do is the diencephalon, and then all we got to do is the brainstem and the cerebellum, and then we'll be done with the brain. So, the diencephalon, if you watched my lab video on the brain, you already know where the word diencephalon comes from because we talked about that in the lab video. Your nervous system, when you were a tiny little embryo, was just like a tube thing. And then that tube thing starts to get some bulges. And it gets 4 bulges. Boom. And this second part in the developing nervous system of the embryo is called the diencephalon because it was the second in the head. It's the core of the brain. The cerebrum, as the cerebrum develops, the cerebrum is going to kind of mushroom and grow around it. So we can't see the diencephalon until we start slicing that brain up. And when we do slice it up, and when we see the diencephalon, we're going to see that it is composed of 3 gray matter structures: the thalamus, the hypothalamus, and the epithalamus. So let's look at all 3 of them right here in this mid-sagittal sliced brain. This purple egg here is our thalamus, right side of the thalamus. This area is our hypothalamus, hypo under because we are kind of under the thalamus. Then back here we have the epithalamus on the outside. We tend to think of epi as on top as well. And if we weren't bipeds, epithalamus would live up to its name better. All right, we're going to go through all 3 of these guys. Let's start with the thalamus. These 2 eggs right here make up the thalamus. It is a big chunk of your diencephalon, 80%. It is a relay station, a switchboard, where sensory signals come in. Here's the deal. Most sensory signals, you want to ignore them because there are so many that it would drive you bananas if you were getting all of your sensory information all the time. So your diencephalon is going to do a lot of pruning and editing. It's going to say, oh, this one's not important. This one's been going on for a long time. We don't need to worry about this one. We're used to that one right there. It does let new and intense stimuli through, and it makes sure that signals go to the right place as well. So it is a sensory relay station. It does have cognitive jobs. I mean, it is a big part of the brain after all. It does have motor input. It has motor input because it talks to the basal nuclei and talks to the cerebellum. So, the thalamus. And by the way, the 2 halves of the thalamus are connected. We saw this in lab by this little oval right here, which is called the interthalamic adhesion. Okay. Here we see just the idea that all these senses are going to the thalamus. So taste from the tongue, purple lines up to the brainstem to the thalamus. Senses from your body, somatic senses, balance, proprioception, touch, to the thalamus. Equilibrium, your senses from your semicircular canals in your inner ear, to the thalamus. Sound from your ear, to the thalamus. Vision from the eyeball, to the thalamus. The only thing that doesn't go to the thalamus is smell. Smell is so important for animals that it needs to get to the appropriate cortex fast, so it bypasses the thalamus. Now, for us, this doesn't really matter, but this is just another one of those evolutionary remnants that we have in our body. Then from the thalamus, somatic senses go to the postcentral gyrus, visual senses to the visual cortex, hearing stuff to the auditory cortex, and so forth. All right, that's pretty incredible. Let's talk about the hypothalamus. I think we're going to save that for next time we meet, okay? Because we're getting on to a half an hour here. You can see if I can do one more lecture on the brain. finish everything out in another half hour, and then we'll be done with the brain. All right, guys, it's been fun as always. Take care. Stay safe. Adios. Goodbye.